Well, nevertheless, let's go ahead and progress through this stuff. Yesterday, we had left off with a drawing that was much like this. And we had established that this was a mitochondria. And without being too specific, because we had been specific yesterday, we had established that this was the reactions of glycolysis that had happened on the outside, right? Which made glycolysis aerobic or anaerobic? Definitely anaerobic. And we were going to go and be aerobic, like the 1980s, basically. Get it? Aerobics? Spandex? Okay, well, whatever. We're gonna go be aerobic inside the mitochondria. So now I need to actually take my next reaction, and I need to take those two pyruvates, and I need to go into the mitochondria. Remember, how many carbons long was a pyruvate?
3.
Okay, so there's 1, 2, there's the third carbon, and since there's 2 of them, I should draw out the second one here. Okay. Now, when we do that, and we actually do this shunt, as I like to call it, that's what I was taught in college to call it. It takes our 2 pyruvates and sends them into the mitochondrial matrix, because remember that this right through here is the mitochondrial matrix. And this process, this shunt that takes your 2 pyruvates in, actually does a couple different things that form 2-acetyl coenzyme A's, 2-acetyl CoA's, from your 2 pyruvates, where each acetyl coenzyme A is 2 carbons long.
How does the other 2 carbons?
Good question. Where are the other 2 carbons going? They're one of 3 things that are happening in this process. The first thing that's happening is you're releasing the 2 carbon dioxides. You're also having to add into it the coenzyme A. See how it's 2 pyruvates and there's the coenzyme A? It's like an additional little molecular piece. And then lastly, I make 2 NADHs from 2 NAD pluses. Now, remind ourselves, just because it's important to do, the NAD plus to NADH, is that an oxidation or is that a reduction? So this is a reduction process that's taking the NAD plus and making the NADH. What does that mean you're doing to the pyruvate? Are you oxidizing or are you reducing the pyruvate? Okay, so this is going to make the overall reaction an oxidation. And remember from yesterday, we had said that what you're doing with your sugar the entire way through is you are oxidizing the sugar. Cool? Awesome. Now my 2 carbon piece, I need to continue to react it, correct? To remind ourselves, we've done a couple of things that are important. We've done glycolysis, you've seen the shunt. So now it's time to go into the mitochondria and actually finish this job. I like to draw out a different picture each time just because it helps you identify the major pieces. So we had done glycolysis. We had done the shunt. And now we're inside the actual mitochondria, inside the matrix. Get it? We're in the matrix.
That was really confusing.
Really?
It confused me. Well, probably because I wasn't really paying attention and didn't really care about it. That's how I thought it was Inception. Yeah, same. I didn't like Inception.
Inception, something else. So you have your 2 acetyl coenzyme A's, right? Which were from the 2 pyruvates. And I'm going to incorporate them into an important cycle. That cycle is the citric acid cycle.
Is that related to the Krebs cycle?
It's the same thing.
Oh,
How many carbons were each of the acetyl-CoAs? So that means that there's a total of 4 carbons still locked up in my sugar, essentially. So this would be a point in time where it's valuable to finish the job. To do so, I'm going to release 4 CO2s. Now I've produced 6 carbon dioxides along the way. We produce 6 NADHs, 2 ATPs, and 2 FADH2s.
Is that FADH2?
Mm-hmm. FADH2 is like the first cousin to NADH. It's another electron carrier. It's another bank account.
And how many ATPs? Did you say just one?
There's just actually 2 ATP. I apologize. Now, at this point, we're essentially done with breaking it down. The sugar is no more by this point. Think about it. The sugar, the glucose you started with was transformed into 2 pyruvates. The pyruvates became 2 acetyl coenzyme A's, and you broke down everything. In other words, going back up to that drawing from yesterday, where you take your high energy glucose and you break it down into carbon dioxide, you've now produced your 6 carbon dioxide. And if you think about it along the way, you pulled off all these H pluses and electrons so that you ended up producing a grand total of 10 NADHs and 2 FADH2s. Bless you.
How many ATPs were produced? Was that just irrelevant?
No, we could include that as well. You've produced 4 ATP. And the mechanism by which you've produced them is a mechanism known as substrate level phosphorylation.
That's with an enzyme, correct?
That just means that the phosphate was on the sugar derivative.
Okay.
The phosphate was on the molecule. You didn't take ADP plus just a random P to make ATP. You didn't add 2 phosphates to 1 to make the triphosphate. The phosphate was actually located on a sugar. So that's why it's called substrate. The phosphate was on the substrate,
Okay.
a reactant.
Where did you say that this occurred? Was this?
All these 10 that are made,
Yeah.
remember in glycolysis you had made 2 NADHs,
Uh-huh.
you had made 2 ATP, and you had made 2 pyruvate, correct?
Yes.
Then you did the shunt to take your 2 pyruvate in and form 2 acetyl coenzyme A from that. Doing that made 2 NADHs, correct?
Uh-huh.
You also blew off 2 CO2. So let's count up. 2 NAD, 4, and 6 makes how many? 10?
Okay.
There's the 10 NADHs. There's your 2 FADH2s, and your 4 ATP come from 2 there and 2 there.
So that's just the whole, like the overview of like glycolysis to the Krebs cycle?
Yes. That's correct. So at this point, we're done breaking down our stuff, our major stuff. Now, do I have a ton of ATP? Made 4. Now, if you read your book very closely, you'll see that glucose should actually yield somewhere along the lines of like, I think 150 or so ATP. That's its max, but that's like maximum. That's with 100% efficiency. Reality is that max efficiency is like impossible on everything in this world, including biochemistry. And so the max ATP you could expect is going to be a little bit under 40 ATP. But even still, how much ATP have we made through this process? Only 4, which is about 10%. That means I still have in the realm of 32, 34ish or so ATP to produce. Agreed? Where do you think I'm going to have this power to produce an excessive amount of ATP? I'll let you guys contemplate it. In fact, that's what I want y'all to do because I want y'all to be able to solve this question. I want you guys to huddle up in a little circle or whatever it is you guys do, and you know, let's have y'all figure this out because I've got carbon dioxide, which is about as pointless as an empty soda can. It's worth about 5 cents in the right state. It's nothing. So I've made some ATP, but I'm telling you right now, this number 4 is 10% of what it's supposed to be. So I want you guys to theorize on where I could find more energy without having to do this process again. Let's say maybe a minute. Work together. transport chain. Kind of the electron transport chain. What do you think the electron transport chain is going to transport?
Electrons.
Duh. Right? So, let's draw this out. This time, though, I'm going to actually take up a large part of the paper. What is this right here?
Mitochondria.
You sure?
Like 99%.
Yeah. Definitely a mitochondria. So let's talk about this mechanism for producing this insane amount of energy real quick. This electron transport chain. First off, do you remember this is the inner membrane, this is the outer membrane, correct? What are the infoldings called? The cristae. So I'm going to intro you to about a couple of major players real quick. The major players I want to introduce you to are your major proteins of the electron transport chain. I'm drawing these unique shapes so that we understand that they're different objects. Very good, by the way.
You missed Curtis. Curtis was awesome.
There's your four major proteins of the electron transport chain. Now, if you had recalled, along the way, and so I'm going to do this ... feature. We had been taking NAD pluses and making them into NADHs. And we did it so that we ended up having 10 of the NADHs and 2 of the FADH2s. Now, doing that would require an input of an H plus and 2 electrons. So this NADH is electron heavy. And by the time we're done with the glycolysis, the shunt, the citric acid cycle, aka the Krebs cycle, you have about 10 NADHs and you have 2 FADH2s. Now, what the proteins of the system are going to work to do is they're going to work to extract out your electrons and your H's so that you can reform the NAD plus, all 10 of them, and the 2 FADs. Now, doing this is going to allow the NAD pluses and the FAD to do what? Recycle. So I can recycle those. I can use them later on. Now, what two things went into making an NADH from NAD+? The H and the electron, correct? The proton and the electron, because H+ is a proton. So that would mean that now I'm carrying my little electrons. There's a little E drawn in right there. I'll put a circle around it to help us see it. This electron is going to be carried by this electron transport chain. There it is at the very end. At the same time, because the electron is now in this chain, you've actually given reason for the H's to end up on this outer side. Think about it. The electron is negative, the H+ is positive. They're in love, like two magnets. You've yanked the electron off, the electron goes wherever the H is, the H sees the electron over here, the H is shunted out in this space. Cool? Now, your electron is being carried. Literally like that game, you know, where you turn, like you have this big object and you turn around to the person behind you and you drop it off to them, and they turn around and they drop it off to them. It's that same principle. This electron is being carried like a 50-pound sack of feed, basically, between the people, or in this case, the protons, or the proteins. Now, in the end, the electron sits here. Now, from chemistry back in the day, you might remember that there are some elements that are electron hungry. The nonmetals are electron hungry. What's a nonmetal that is a part of the cell respiration reaction that I haven't brought up yet. I'm going to give you like maybe 30 seconds. You can work together as a group. I want you guys to think of what is the non-metal that I said was a part of cell respiration, but I haven't brought it up yet.
That's a good question.
Good question. So we have oxygen, right? Oxygen is very electronegative. So what does it do? Steals electrons. Now what happens to the charge of oxygen? Yeah, it's very, very, very negative. It's negatively charged oxygen. What is something negative attract? Something positive. The opposite is attracts. Where's the positive at?
The hydrogen.
It would like to get in, but the problem is that there's no doorway, correct? So you've seen now the setup of the electron transport chain. Now it's time to come over here and show you chemiosmosis / oxidative phosphorylation. We left off like 25 seconds ago with the H pluses want to come back in, right? They need a doorway. You might be thinking that I just made a door randomly, but it actually exists. That door is an enzyme. Its name, ATP synthase. I know it's an enzyme because all enzymes end in ASE. What do you think the ATP synthase lets in? It lets in your hydrogens, right? Why do the hydrogens want to go inside? Because they're against, or they're going to need to go down, their electrochemical gradient. What two pieces make up an electrochemical gradient? I'll give you a hint. It's in the name. Electrical and then chemical. There's a lot of H's on the outside in this space. Where do they want to go? The inside. There's a lot of positive in this space. Where does a lot of positive want to go? To the negative on the inside. Electrochemical. Now when the H plus ends up down here, what do you think it combines with? And what do we have formed? Wasn't that another product of respiration? Weren't your two products the CO2 and the H2O? Now you know where they're both formed.
Do they just kind of like rip apart the oxygens from each other?
Mm-hmm. They do.
Like, how
So
does
that
it happen? Just like, oh, bye-bye.
it's just it's basically I think they're going to split up actually somewhere like right through here. As they become very negative, I suspect they break up.
Okay.
And I think they can do that because at that point, both oxygens are going to have, because you have a ton of electrons being shunted through here. See, when O2 is O2, it's together, these two oxygens are together to share electrons to facilitate a lacking in their life, or in their valence electron shell. Yeah, the diatomics, they exist like that. But once you add electrons to it, they don't need to hang out anymore. So it's actually going to be oxygen that's just by itself very negative. In fact, each oxygen is going to be 2 minus in charge. And each of these oxygens could give away an electron to hydrogen. So 2 H's come in for every 1 O2 minus, and you have 1 water formed.
That makes sense.
Now, what do you think is happening in this enzyme? The H pluses are going through, right? But imagine like, and I'm horribly bad at explaining this part, but it's like what is it called? Not a windmill, but you know when you let the water rush over it?
Yeah.
Like a turbine, basically, right? So like imagine that the H pluses land on this turbine and it spins it around. Well, the spinning action basically allows you to produce some ATP from ADP, an inorganic phosphate. Now, where do you think you get and then you're like, well, this seems so illogical. Where are you getting your ADP from and your phosphate? What are you doing right now? Hopefully.
Breathing.
And what else are you doing?
Taking in oxygen.
No, I want you to tell me like just literally what are you doing right now?
Sitting.
Sitting, resting?
No.
Using energy.
How?
With our
Just staying awake.
Just the fact that you're thinking uses energy.
So you're
So this right here is you. right? How am I ever going to get back ATP so I could think again tomorrow? What do we do to refuel?
Eat food and drink.
We do this process with a goal that if I can combust it the right way and be inside my mitochondria, I can make this excessive amount of NADH. I could pull off its electrons, pull off its hydrogens, let them flow back through, and what do I remake at the end of the day?
ATP.
And that's if I can do the combustion with my oxygen. Because remember, off to the side from yesterday, if you never go inside the mitochondria, you produce basically not enough ATP. And you're like, Mr. Fields, how do you know this? Because when people don't breathe, they die. Right? What do we think?
How much ATP is produced from the ATP synthase?
Amazing question. About 34. And a good way to remember that, and this is a cheap way, but it works well enough. For every NADH, it actually can result in about 3 ATP. And for each FADH2, you could get about 2 ATP. How many NADHs had we made through the process, the overall process? 10. What is 10 times 3? And what is 2 times 2 for the FADH2? 34. So there's your 34. How many ATP did you make back in glycolysis and Krebs, grand total? About 4. So 4 out of 34 were by substrate level, and 4 out of 38 were by substrate level. 34 out of 38 were by oxidative phosphorylation. Oxidative means that I had some oxygen there, right? I continue to oxidize my sugar. So what do you think? What do you think about breathing now?
It's very
It's fun.
Yeah, it's fun. Without it, we would never have progressed through bacteria. See, the deal is that once upon a time, about 2.7 billion years ago, oxygen began to accumulate in this atmosphere. Now, oxygen is actually a pretty negative chemical. It's fairly reactive, and it can allow, it's just not helpful for biology in a lot of cases. But, so there's a lot of creatures that were harmed by it. But is this amount increased? And how did this increase? Well, that's a great question, because you had the first startings of photosynthesis in bacteria, the cyanobacteria. So as the amount of O2 increased, you had to have a mechanism of using it and producing CO2 so that the cyanobacteria had a way to work. And so the circle of life, not that song from The Lion King, I mean the real circle of life, not that waste, that's where it began, about 2.7 to 2.4 billion years ago, according to evolution at least, was this process had its inner workings. But this actually came second. Photosynthesis came first. Cool? Fairly manageable? Yes.
This might be a real stupid question,
There's no stupid questions.
we'll see. But what about all that other stuff in the air? Because oxygen is only a small portion of it, so what
The-
happens to the other stuff?
The most largest piece, or the most, that's not good. The largest piece, aside from oxygen, is going to be the nitrogen. And what happens with the nitrogen that's in the air is it's basically cleaved. You break this bond right down the middle, so you have two little nitrogens that are no longer connected. You combine them with hydrogen, and so you're combusting with hydrogen essentially, and you actually produce something called NH3 or NH4 plus, or you can even combust with oxygen to make NO3 minus. You basically take the nitrogen from an unusable diatomic form, a clobber fin as you call it, and we make it into usable form. Now, actually, we don't do that. Well, we do. We do. Bacteria do it better than us. Lightning will do it as well. Or we can do it, but to cleave this bond, because it's triple bond, it's extremely expensive. The reason we use nitrogen in this case and we cleave the bond, or bacteria cleave the bond, is because we know plants need nitrogen. It's called fertilizer. And bacteria fertilization, if you will, this nitrogen fixation is the best way to do it. Now, in the event that that's not possible, and we have to go buy fertilizer from Lowe's or Home Depot or, I don't know, wherever else you can buy it, then that means that there was a manufacturing company that cleaved this bond, split them apart. So that's where the nitrogen's used. Your fun fact for the day is that we use 10% of the world's oil reserve each year to facilitate the breaking of this bond. Where bacteria could do it for free. We're so wasteful. The other stuff in the air, like the carbon monoxide, it's not a large amount. The carbon dioxide, of course, is produced through this process and is fed to plants. In the higher levels, there's tons of O3 minus, or O3, which is ozone. and that's the actual like compound that helps to block out UV rays and maintains the layer, if you will, that is the sunscreen for this planet. That's your major players in the atmosphere. The other gases that are there, like a lot of them are either by accident, or like in other words, they're pollution, or they don't really play a pivotal role. They're more of a filler. Cool. What do we think about this? This is pretty manageable stuff, right? One thing that I think really helps out is definitely one, reading the book, reading the cliff notes, and drawing this out. This is amazingly complex, and I would encourage everyone to kind of spend some time drawing it. You learn a lot.
The book has a really cool picture that shows like all three of them together.
Yeah,
It's
and
really simple.
I've got that picture here. I'll show it to you real quick. The picture I think we're kind of looking for a little bit. I think it's this one.
Do we have an online textbook?
Mm-mm.
Oh, yeah.
Is this the one you're talking about?
Yeah.
The one thing I don't like about this one is it doesn't include the count, but we can go through it real quick. There's how many electrons made through or how many NADHs made through glycolysis? 2 through glycolysis. How many FADH2s are made from citric acid cycle? 2. How many NADHs are made from citric acid cycle? 6. 6. Where are the other 2? So I'm supposed to have, and everyone keeps saying 10, right? 10 NADHs are what are shunted into this phase. 10 NADHs show up here. 2 from here, 6 from here. The other 2
Oh, are
from the shunt, from the in-between state. 1 glucose, 6 carbons long, becomes 2 pyruvates, each 3 carbons long. The 2 pyruvates become 2 acetyl coenzymases with each 2 carbons. I'm now down 2 carbons. I blow them off through carbon dioxide right through here. How many ATPs did I make in glycolysis? It's a trick question. You actually made 4, but what do you net out?
2.
Because you invested 2 initially. How many ATP do you make from a citric acid cycle? 2. You made 4 out of the total of 38, which means your other 34 came from the oxidative phosphorylation through chemiosmosis in the electron transport chain. Cool? Very, very good stuff, right? We got this. Destroyed. Awesome. That's what I like to hear. Little biochemists.
Can I ask you kind of like
Ask me.
question?
I'm ready.